All right. Well, good morning, everyone. I'm your opening act for these superstars. So welcome to our Biotech Connector series. Welcome to Frederick National Lab, both folks in person and a lot of folks online. My name is Vladimir Popov. I'm the Chief Innovation Officer here at the Frederick National Lab for Cancer Research. Biotech Connector event, it's a collaboration between Frederick National Lab and the Frederick County Chamber of Commerce. This event is kind of exclusive quarterly speaker series that started a long time ago, actually. I've been here for 9+ years. Started way before that. Then it took a little pause. We restarted it again and then took a little pause again during COVID. And now Luba has been doing a great job organizing this event and bringing some excellent speakers. So the idea for this event is to bring experts from our surrounding area. So this is Frederick County and regional event as well. And we always bring one of the experts from our own lab. And the event usually focuses on specific area of research. Basically it's 2 to 3 speakers every time, and we try to wrap it in about an hour or so. And I'm very happy to see a lot of people coming back to be in person here. So Frederick National Lab, for those who don't know, we're a collaborative enterprise. We're a national lab that focuses on cancer research and infectious diseases and emerging diseases and HIV and AIDS. We have existed for good 50+ years, and as a national lab, we're a national resource, and therefore we collaborate heavily. So if you see something you like from here, please approach us. We have folks from Partnership Development Office here. Luba is one who's organizing this. Amanda's sitting right here. Victoria just walked in. Sorry, Victoria. We also have our leadership here, Dr. Nathan Bintrovsky, who's the president of the lab here. We have our chief science officer here. So feel free to collaborate, to ask questions after that, and to stay for networking. So some of the things to cover this morning. First and foremost, there's coffee and there is food here, so please help yourselves. Restrooms are across the hall. When the speakers will introduce the speakers, and after each speaker, you will have a chance to ask a few questions. Raise your hand. You can walk up to the microphone. You can just stand up. For folks online, you can unmute yourself and ask questions. We'll try to move this in a efficient way. So we'll have also a networking after this, so you will have a chance to speak to your colleagues. So let me move on to speakers. We are joined by 3 very distinguished speakers who will share recent advances in liquid biopsies. We have, like I said, one of our own, and we have 2 very big experts from the outside. So I'll introduce all 3 speakers. Kaitlin Victor, sitting right here. Hi, Kaitlin. She's an associate scientist at Labcorp Oncology. She grew up in Southern California and went to college in University of California, Riverside. She later attended Johns Hopkins University School of Medicine and received her master's degree in human genetics and molecular biology. Kaitlin's thesis involved 2 related mitochondrial disorders, methylmalonic acidemia and propionic acidemia. She has also worked on a DoD doing dengue vaccine work, and in 2021, Kaitlin moved to her current position at the Personal Genomic Diagnostics, PGDX, which was later in 2022 acquired by Labcorp. She's working there now on the assay development for a minimal residual disease assay. We have Amanda Peach, as I said, one of our own, and she's a Research Associate 3 at the Molecular Characterization Lab, also known as MOCA. This lab is here at the Frederick National Lab. Amanda currently is leading the liquid biopsy group at MOCA. She's focused on advancing non-invasive diagnostic tools that leverage circulating biomarkers for early disease detection and monitoring. Their work plays a critical role in bridging cutting-edge molecular technologies with real-world clinical applications. Amanda holds a master's degree in biomedical science from Hood College and second master's degree in bioinformatics from Johns Hopkins University. This reflects a strong interdisciplinary foundation that combines laboratory science with computational analysis. Really, just 2 masters? No third one? Okay. And last but not least, our third speaker is Dr. Lorenzo Rinaldi, who will be joining us virtually because, believe it or not, Dr. Rinaldi is in Africa right now. So thank you for joining us virtually and fighting the time difference. Dr. Rinaldi's actually wife is a diplomat, and I don't know if I was supposed to say this, but it says here. So he is the head of Translational Science and Scientific Alliance Manager at Delfi Diagnostics. Dr. Rinaldi has received his BS degree in biology from University of Florence, Italy, after which he spent a year working in malaria research as an assistant scientist at Harvard Medical School. Dr. Rinaldi earned his master's degree in biomedical research from University of Pompeu Fabra in Barcelona, Spain, followed by a Ph.D. in epigenetics and cancer initiation from the Center of Genomic Regulation and Institute of Research of Biomedicine in Barcelona. After receiving his Ph.D., Dr. Rinaldi was a genetic lecturer at the University of Lesotho, South Africa, for about 6 months, and then he moved to a postdoctoral position in NCI Bethesda. He joined Delfi Diagnostics as a senior genomic scientist, when later he became the head of Translational Science. So, impressive lineup of speakers. Please enjoy the talks, listen, and ask questions. So, we're going to welcome our first speaker, Kaitlin Victor.
Okay. Good morning. Here's my presentation. So as I was introduced, my name is Kaitlin Victor. I'm a scientist with Labcorp Oncology in Baltimore, and I've been there since 2021. And I have been working on our minimal residual disease, our MRD assay, since I started in 2021 when it was BGDX. So I'll be talking about our MRD assay, along with just some basic info about liquid biopsies in cancer diagnostics and surveillance. So the agenda for my presentation is I'm going to start with the liquid biopsy background, then I'm going to give some background on our Labcorp Plasma Detect assay, which is our MRD assay. I'm going to talk about our CLIA validation of LPD at the Baltimore site, and talk about how LPD was used to look at the Provence study. So we'll start with the background. So as a lot of you will probably know, 2/3 of patients diagnosed with solid tumors initially present with local regional disease and potentially are cured using surgical or therapeutic intervention. Now, there's a subset of patients who have residual tumor cells remaining post-surgery or post-therapeutic intervention, which potentially leads to disease recurrence. And these residual tumor cells can be detected using or through circulating tumor DNA analysis to capture our MRD. Using liquid biopsy-based MRD analysis, disease relapse can be detected earlier than clinical presentation or standard of care, radiographic imaging, allowing physicians significantly lead time to intervene with subsequent therapy. So, circulating tumor DNA is what enables our liquid biopsy applications. We've had major advances in our ability to detect tumor-derived material in blood and other biofluids. Sensitive assays can detect cancer-specific analytes like ctDNA, proteins, mRNA, etc. And ctDNA is composed of small fragments of nucleic acid that are not associated with cells or cell fragments. And the clinical application for ctDNA has been termed our liquid biopsy. And ctDNA-based assessments can inform the detection and genomic landscape of tumors. And as I mentioned in the previous slide, liquid biopsies have the potential to detect, characterize, and monitor cancers earlier than is possible with our current conventional approaches. So I'm going to define MRD using the NCI definition. So it's used to describe a small number of cancer cells that remain in the body during or after treatment. MRD can be found only by highly sensitive laboratory methods that are able to find one cancer cell in millions of normal cells. And seeing if there is minimal residual disease can help plan treatment, can tell whether or not treatment is working, or if cancer has come back and help make show prognosis. And ctDNA MRD detection following our definitive interventions such as surgery or adjuvant chemotherapy predicts relapse. It can also help identify candidates for adjuvant therapy post-surgical resection and measure response to each of the treatments. Things that impact MRD or our ability to detect MRD are assay type, the amount of ctDNA being released by the tumor, and technical and biological background that all affects our MRD results. This could revolutionize personalization of adjuvant and consolidation therapy in cancer. This, excuse me. So basically, I wanted to show where our Labcorp liquid biopsy assays fall in our cancer diagnostic continuum. So Plasma Detect is our MRD assay, and that currently is being used post-resection or post-ACT to see if there is ctDNA still circulating. We also have Ilioplasma Focus and Ilioplasma Complete, which are also liquid biopsy-based assays. But those are more commonly used post-clinical recurrence to look and see what additional mutations the tumor may have picked up. Are there potentially clinical studies that these people could be enrolled in? Are there other immunotherapies that they may now be eligible for? So those are kind of post-resection, but our Plasma Detect MRD assay is for like earlier post-surgery, post-ACT. So now I'm going to discuss the LPD assay and give background on how ours works. So Plasma Detect and MRD. Our Plasma Detect assay is a whole genome approach. We use plasma, FFP tissue, Buffy coat, like I said, in an integrated whole genome approach. It does not require a bespoke patient specific panel to be designed. It has 10x improvement in informative reads compared to a bespoke approach, which normally focuses on 16 mutations. This enables us to also have a turnaround time for prospective testing of less than 10 days. We have a decentralized global platform called the Elio platform that we already have a framework for that we could potentially use for LPD. And LPD provides additional genome-wide insights that we wouldn't see if we just did an exome-based assay. It also is more informative for longitudinal studies and hopefully earlier detection of recurrence. And so this whole genome sequencing method provides a more sensitive and specific ctDNA status with a faster turnaround time. This is our tumor-informed LPD workflow. So this is our current LPD workflow. We have our sample accessioning, so our tumor tissue, our plasma or white blood cells. We process in library prep, and then we sequence on a NovaSeq. And once it's done sequencing, we have an analysis, we analyze the data, we have a report generation, and we review. We take out the time-consuming bespoke approach where you make a bespoke panel specific to the patient and then resequence. So it's a simplified workflow by removing the bespoke panel design. What is also nice, that because it's a whole genome sequencing approach, is we are able to identify thousands of high confidence patient-specific somatic alterations that you would miss if you just did exome. And on the left, you basically see different cancer types and the number of high confidence SNVs you would expect or you expect to see. And then on the right, we have, again, just kind of our how our assay works. So you have your whole genome sequencing of the tumor tissue, the white blood cells, and the plasma. You have demultiplexing, pre-processing alignment, and variant calling, which gives us tumor variants, matched blood variants, and cfDNA variants. And we compare our matched blood variants to our tumor variants to basically filter out any germline or clonal hematopoiesis variants. So we just have a candidate somatic SNV set, along with our candidate cfDNA SNV set. And both of those are then put into our ctDNA machine learning algorithm, and that produces a ctDNA status and a level percentage for us. So we can call whether or not someone is ctDNA positive and at what level. Okay, so now I'm briefly going to talk about our CLIA validation of LPD at our Baltimore site. And there's a lot on the slide, but the most important things is one, our CLIA validation of LPD met all of our acceptance criteria. But additionally, the most important two spots are our analytical specificity and analytical sensitivity. So for our analytical specificity, we use non-cancerous plasma samples, and we ran them against a set of known tumor samples. And we weren't calling too many false positives. So our analytical specificity is 99.4%. For our analytical sensitivity, we made 3 contrived cell lines and ran them at different levels. And these included a breast cancer and a melanoma cell line. And our analytical sensitivity for our assay is 0.005%, which is lower than the Natera MRD assay, which is 0.1%. In addition, we have our precision, repeatability, reproducibility, and our analytical accuracy, which again met our acceptance criteria, showing that we had a validated assay that performs as expected. And this gives us, or tells us that there is significant potential for tumor-informed, non-bespoke, panel-based MRD approaches for ctDNA detection across a broad range of solid tumor types and curative intent clinical settings. This independent validation data demonstrated there's adequate analytical performance of LPD in a CAP CLIA lab for pan-solid tumor research use, together with investigational use in early stage colon cancer to support the interventional MEDOC CREATE clinical trial. Also, right now, LPD, we are running early access LPD program at the Baltimore Labcorp Oncology site, and we're running clinical samples. So now I'm going to talk about using LPD to look at the Provence study. What the Provence study was, it was a study to investigate the prognostic value of post-surgical ctDNA testing for disease recurrence in adjuvant chemotherapy-treated stage 3 colon cancer patients. It was an observational study with the prospective Dutch colorectal cancer cohort. There was 26 participating hospitals with 236 patients enrolled between December 2016 and October 2021. They are stage 3 colon cancer patients who have had adjuvant chemotherapy, and there is a post-surgical blood sample available. And what we did is we analyzed 1 time point post-surgery, 1 time point pre-surgery, and 1 time point post-ACT, which was analyzed for the study. So what we have here is our ctDNA detection in the pre-surgery sample, which was 93.4%, which you can see here. Which is what we expected, since you would expect a stage 3 colon cancer tumor to be shedding a decent amount of cells and ctDNA. And then what we have here is we looked at time to recurrence in all patients, and then time to recurrence in patients that just recurred, and whether or not that could be stratified based on whether or not a patient was ctDNA positive or ctDNA negative. And what we found is that if you had positive ctDNA post-surgery, it's associated with a higher risk of recurrence. And you can see that in the Kaplan-Meier curve, oh, there is no pointer. Sorry, I point with my hand and this might not be helpful. But the top in red, those are a ctDNA negative cohort. And then the teal color is our ctDNA positive, and you can see there's a significant difference between recurrence in our ctDNA negative population versus ctDNA positive post-surgery. And for those patients that recurred, the ctDNA positive patients experienced a shorter time to recurrence than the ctDNA negative patients, as seen in the second Kaplan-Meier curve. So what this tells us is that post-surgical ctDNA testing improves the stratification of stage 3 colon cancer patients for disease recurrence and complements our existing clinical pathological risk factors. So in summary and conclusion, liquid biopsies have the potential to detect, characterize, and monitor cancers earlier than is possible with conventional approaches. The Labcorp Plasma Detect integrated whole genome sequencing method provides a more sensitive and specific ctDNA status with a faster turnaround time. And analytical results for specificity, sensitivity, and accuracy met expected criteria with high specificity and sensitivity. And detectable minimal residual disease post-surgery is associated with recurrence and suggests there is a benefit for ACT intervention in those individuals. And then that's it. Are there any questions? Yes.
Yeah, so for the whole genome sequencing, can I ask the coverage of your, generally?
Yes. So generally for our tumors and for tumors, we want coverage of 30x, and for plasma, it's 20x right now in this iteration of the assay. We are working on another iteration of the assay where we're looking for higher coverage. But right now it's 30 for tumor and 20 for plasma.
Same as ctDNA.
Yes, yes. No problem. Yes.
I was curious in the clinical experiment that was done. Let's see the point of persistent
Yes.
signal or prognosis. I was wondering if your assay would detect emerging mutations that are actionable.
So we haven't looked at that, but yes, because we get the somatic SNV mutational profile, if you were to take longitudinal time points, you could follow what mutations you're seeing and if mutations are changing, and that could give us more targets for treatment. We haven't done that yet, but I feel like that is probably something we do want to look into.
If I could add,
Mhm.
this is more of a spinal oncology.
Okay.
So, the always filtration.
Yes.
You may actually feel what's in it.
Yes.
Because there's increased evidence. There's sort of the two-part plus. This sample was and these three communications, there's a subset of patients with system positive simply because they're having.
That is a good question. I don't remember off the top of my head how quickly post-surgery we are taking the first time point. I know it is in some of we have that information. I just don't remember. But I can let you know. And also we do see patients that are persistently ctDNA positive over all of these time points that we looked at. And I do think generally their prognosis was worse than the people who either were ctDNA negative the entire time or vacillated. So we had some patients who were ctDNA negative and became ctDNA positive, or were ctDNA positive and then became ctDNA negative post ACT treatment. So I can get you the information. No problem. Yes.
So great, thank you.
Thank you.
I think one of the challenges that emergence space face, and I want to false negatives,
Yes.
actually being positive, but they're actually not simple.
Right.
And I appreciate all of the validation. It would have been probably really nice to have even more separation.
Yeah.
Had been said between positive and negative patients. So we had arguments of reverse sequencing, that really question.
Yes.
Plasma, it might be able to get there. wondering specificity study.
Mhm.
To demonstrate high specificity, the tumor positive patients, were those early stage or were they late stage?
They were later stage. Yeah.
Early stage, okay.
So they were, yes.
The late stage ones are not going to tell you how specific you are.
Mhm.
Normal, not technical.
Right. Yes. I believe they were stage 2 generally, that we used for the validation. If I'm not mistaken, I believe they were mostly stage 2 with some stage 3. No problem. Yes. So for, oh, you know what? I didn't have a slide that discussed that. Just realized I took that one out. So for ctDNA, we input 10 nanograms. For tumor, it's 200 nanograms, and for our whole blood cell Buffy coat, it's 50 nanograms. And that is based on qubit for this assay for LP. If there are no more questions, I will hand it over. Thank you.
So good morning, I'm Amanda Keach. I'm a Research Associate 3 at the Molecular Characterization Lab with Frederick National Labs. I will be presenting on liquid biopsies in support of NCI precision medicine initiatives. So first, what is ctDNA? Cell-free DNA or cfDNA are fragments of DNA that's found circulating in the bloodstream. Circulating tumor DNA or ctDNA are DNA fragments that are released by tumor cells into the bloodstream. So this figure illustrates how tumors release different biomaterials into the This figure illustrates how tumors release different biomaterials into the bloodstream. So there's secretion, necrosis, and apoptosis that can release cfDNA, ctDNA, or circulating RNA. Tumor cells can also be released into the bloodstream through shedding-like processes to release circulating tumor cells or CTCs. And endosomal vesicle processes release exosomes, which can introduce nucleic acid and proteins into the blood or lymphatic system. So why is liquid biopsy important? There are multiple advantages of liquid biopsy over solid biopsy. First, it is minimally invasive, which ensures patient comfort and is less painful. It also reduces the need for a repeated tumor biopsy. Next, it provides a molecular landscape of the tumor with prevalence mutations, and it also captures tumor heterogeneity. It enables frequent tumor monitoring and treatment assessment. So you can assess a patient to treatment by monitoring changes in ctDNA fraction over time. So you can track tumor evolution and uncovers the potential resistance mutations. You could test that initial screening, treatment progression, and treatment resistance. And one limitation of liquid biopsy is the detection of ctDNA is highly dependent on the degree of tumor shedding. The NCI ctDNA V2 assay supports the NCI clinical trials. It is a 523 gene panel that identifies SNVs, CNVs, indels, 23 fusion drivers, blood microsatellite instability, and blood tumor mutational burden. Our assay input requires a minimum of 10 nanograms, but we can go up to 40 nanogram input. It also includes genes associated with clonal hematopoiesis of indeterminate potential, or CHIP. CHIP is when blood stem cells acquire certain mutations, but the patient is not diagnosed with a blood cancer or blood disorder. It was validated for research use only, with extensive testing with over 300 libraries tested towards validation of version 1, and then a bridging study was done to bridge to version 2 of the assay. The primary primary application is for retrospective analyses in support of NCI trials, such as NCI match, investigating mechanisms of resistance and response, and exploring early changes in ctDNA fraction as potential predictive biomarkers. It is being developed in a CLIA lab and may eventually be clinically validated for treatment selection in the future. So on the left side of this slide, we're looking at how we extract our cell-free DNA from plasma. So we use the Chi Symphony for this extraction. So first, the plasma is mixed with binding buffer, proteinase K, and magnetic particles. And then the cfDNA binds to the magnetic particles, and it then undergoes magnetic separation, and contaminants are then washed away. The final step is elution into the matrix tubes for pure high-quality cfDNA for downstream analysis. So on the right is our quality control check. We use the Agilent tape station. So here you can see the electropherogram that's outputted that illustrates the DNA fragments. So the fragments should be between 50 and 700 base pairs in length, and at least 50% of the sample should fall within this range. So here you can see there's a mononucleosomal peak and a dinucleosomal peak, which is usual for ctDNA. Anything that is beyond the 700 base pairs is considered high molecular weight and is likely contamination of genomic DNA, and this can come from the breakdown of white blood cells. So in this graph, you can see the yield in nanogram per mil of plasma for a subset of histologies. And the data shows a clear difference in plasma DNA yield across the different histologies, which reflect the variability in tumor shedding. So salivary gland cancer, or SGC with the green dots, show consistently low yields, which suggests limited tumor shedding. In contrast, adenocarcinoma of the esophagus, or EAC with the blue dots, shows moderate yields with a few outliers. And cholangiocarcinoma, or CCA, with the red dots, have moderate yields with quite a few that have high outliers. And this is indicative of substantial shedding in a proportion of the patients. And small cell lung cancer, or SCLC, in the purple dots, had the highest yield overall, which would indicate that it was the strongest shedder. And this makes sense because small cell lung cancer is a progressive and histology. So the NCI ctDNA version 2 workflow is similar to any probe-based library prep workflow process. The process takes 2 days, and the libraries are then sequenced on the 3rd day. So the first step is end repair, where uneven DNA fragments are repaired to generate blunt and even ends. Next is A-tailing, where a single adenine base is added to the 3' end of the DNA fragments. Next is the UMI containing adapter ligation. UMI containing Y adapters are ligated to DNA fragments, and these unique molecular indices or UMIs tag each strand of cfDNA and are introduced before any PCR amplification. This enables accurate error correction downstream. The ligated libraries then undergo purification, followed by the addition of indexing primers. Next is hybridization and capture. So probes hybridize to the target region of interest, which are then captured and enriched using syntavin-encoded magnetic beads. The non-target regions that did not bind to the probe are then washed away, leaving behind only the enriched libraries. The enriched libraries are then PCR amplified, purified, and prepared for sequencing by normalization. B-based normalization ensures a balanced library prep input for sequencing. So once we have the normalized libraries, it then is loaded onto a NovaSeq 6000, which runs for about 48 hours, or the NovaSeq X Plus, which runs for about 24 hours. So a little bit of background on the NCI Match plasma correlative study. So NCI Match was one of the largest precision medicine platform trials sponsored by the NCI. In the central screening phase of the NCI Match clinical trials, patients with advanced cancer underwent screening of their tumor tissue with Oncomine Comprehensive Assay version 2 to determine study eligibility. About 60% of patients had rare or uncommon cancers, and about 40% had common cancers. And the majority could not be assigned to a treatment arm due to study-specific biomarker and histology-specific eligibility rules. Analysis of ctDNA from less common or rare cancers may provide insight into the genomic landscape of these understudied patient populations. This study is designed to evaluate the utility of ctDNA testing in this patient population as a screening assay for future platform clinical trials. So this diagram shows how patients were grouped in the study. So we started with over 6,000 patients enrolled in the NCI match screening phase. Most of them had tissue samples that could be tested at 5,540, while a smaller group did not, 850 that were put into cohort 6. From there, the patients were further divided into whether they were enrolled in treatment arms for cohorts 1, 2, and 3, plasma available for patients with common cancer for cohort 5, and plasma available for patients with rare and uncommon cancers, which is cohort 4. So today I'm going to focus on cohort 4. It includes 2,286 patients with rare and uncommon cancers. And this group is critical because, again, rare cancers are often underrepresented in clinical studies. And altogether, nearly 2,200 patients had plasma ctDNA tests that could be evaluated in cohort 4. And so this slide explains how the NCI ctDNA B2 analysis was performed. So it looks for very small amounts of changes in ctDNA, down to 0.5% variant allele frequency for SNVs and indels, 1.3-fold change for amplification, and 1% allele frequency for fusions. And you can also detect markers like tumor mutational burden and microsatellite instability. On the make sure the results are accurate, the tests are filtered out for background noise, and this is done by removing common harmless variants found in the general population using the NOMAD database. So we filter for less than 0.1%, as well as clonal hematopoiesis variants using a gene list-based approach. And here you can see the CH variants or genes that we filtered out. And on the right is a diagram that shows how results from the ctDNA test were compared to tumor tissue testing. So both the tumor tissue and the ctDNA were annotated by OncoKB. And then the overlap between the two is checked to see how well the ctDNA test matches the tumor tissue test. And then we also get a reliable genomic profile of a patient's cancer without the need for invasive tumor biopsy. So I'm only going to show the cholangiocarcinoma data to give a sense for the data we obtained and the different histologies in this cohort. So on the left, you can see an oncoprint showing the genomic profile of this histology. At the top, you see the median exon coverage, or MEC, followed by the blood tumor mutational burden, and then blood mutational or microsatellite instability. So the purple bars represent microsatellite instability high, and then gray represents microsatellite stable. Below that is the IHC test data by tissue for MLH1, MSH2, and P10. So the red is when it's negative, yellow positive, and gray is when the IHC data is unavailable. And below that is the SMB and CNV data. So the blue triangles represent a tumor variant that is seen in tumor, and the red triangles represent a variant that is seen in ctDNA. So when a square has both the blue and red triangles, it's concordant between both tests. And then the black triangle here represents a copy gain that was observed. So here you can see that there's 7 patients that were identified as microsatellite high only in ctDNA by the purple bars. And there's 1 patient that was identified as mismatch repair deficient by IHC, as you can see by the red bars, in MLH1, MSH2, and P10. But it was microsatellite stable in ctDNA. And there are 2 patients identified with P10 loss of expression in IHC tissue by this patient and this patient, but it did not meet the P10 copy loss threshold in the ctDNA test. Also observed in this histology, but not shown here, was FGFR2 fusions that were seen in ctDNA of 8 patients. So this slide explains the maximum somatic allele frequency, or MSAF. So MSAF can be used as a surrogate for tumor fraction in the blood. Since tumor cells release DNA fragments into the bloodstream, this measure helps us get a sense of tumor burden without a tissue sample. So this metric looks at variants that are known to be linked to cancer. It picks the variant with the highest allele frequency of known cosmic variants present, greater than 50 counts in cosmic. And if that is not available, then a competent somatic variant identified by significantly shorter fragment size is chosen. And if neither of those available, it's the fourth highest allele frequency if the previous conditions are not met. So on the right, we see a plot that shows the relationship between MSAF and how well ctDNA testing matches with tumor tissue testing. The red dots represent patients with high concordance, and the purple dots represent patients with lower concordance. We see that patients with higher concordance also tend to have a higher MSAF value. The median here is 4.2%. When concordance is lower, the median MSAF is down to 0.77%. So in simple terms, the higher the tumor fraction as measured by MSAF, the more concordance between liquid biopsy and tumor biopsy. So this slide shows how well ctDNA testing matches the results from the traditional tumor tissue test across 5 different histologies. So overall, the blood test matched the tumor results 85.5% of the time. The positive percent agreement varies within the individual histologies. The highest match is small cell lung cancer at 98.1%, and the lowest is pancreatic adenocarcinoma at 78.7%. So focusing on pancreatic adenocarcinoma, since it had the lowest concordance, there were KRAS mutations were detected in 225 out of the 232 patients within this histology in the tissue test. 187 of these 225 KRAS mutations were detected in the ctDNA for a positive percent agreement of 83.1%. The median MSAF, using as a surrogate for tumor fraction, was much higher in cases where KRAS was detected at 3.28% compared to cases where it was not detected at 0.29%. And this suggests that the test is more effective when the tumor fraction is higher. So in conclusion, liquid biopsies are minimally invasive and captures molecular landscape of tumor. In this particular cohort of specimens, 85.5% of the oncogenic mutations observed by tissue NGS test were also identified in ctDNA using the NCI ctDNA assay version 2. Liquid biopsies can be used as a tool to investigate mechanisms of resistance and response in real time, and could potentially be used for patient screening or stratification in future NCI-sponsored precision medicine trials. So I would like to thank Chris, Vishu, Val, and Tom for supporting this project. I would also like to thank the bioinformatic team, Rini, Lily, Porva, and Peter for the help with the pipeline and analysis. And I would also like to thank Vishnu, Eric, Brandy, and Lisa for their help bringing this project to completion. Thank you. Any questions?
Great talk. Thank you. For microsatellite instability testing, how many markers you use? That is from NGS analysis FS. There's one case you mentioned that have the mutations and SH2 and LH1, but we do not detect those microsatellite unstable cancer. Do you think that the panel of biomarkers that for microsatellite instability in your panel may not be sufficient, or what might be the cause?
So for the microsatellite instability with our assay is based on the SUM-JSV score. So right now, if it's above 0.08, it's considered microsatellite instability high. So there could be, might need to look into that a little more. Yeah.
I know that the NCI match is largely a study of people with cancer, but I'm wondering whether in your cohort you had those patients with cancer from inherited syndromes like we found in or BRCA mutations, and whether you could detect early on whether those at-risk population had become those with cancer, that would have a huge positive effect in those patients who worry that they're going to have a cancer that arises, and this may go on for decades for them. So I was curious whether those patients were studied in the same match. Yeah. Yeah, so they were patients who going on 29. Identified part of what I mentioned. That
Yeah, so I guess this was similar in vein. So this is a trial that has completed.
Any other questions? Thank you. Okay, Lorenzo, you can go next and share your screen.
It's good? Awesome. Awesome. I think you need to turn around, Luba. Turn around to the camera. All right. Good to see you. Thank you so much. Awesome. All right, let's try to do it. I apologize. It's my mistake that I'm not there. Unfortunately, I live in. So you can still see my screen, I guess. Okay, so you can still see the presentation, the Delphi Diagnostics. Okay. So hello everyone. Good morning, everyone. Good afternoon. A pleasure. Thanks for the invitation. It's a pleasure to be here and discuss liquid biopsy opportunities for both cancer detection and therapeutic monitoring. And you're probably aware we don't see the presentation online. Okay, maybe gone away. Okay, thank you. Let me see. Okay, now probably got away. Okay. Okay. Okay, let's do it again. What about now? Can you see now? Yes, awesome. Thank you so much. Thank you so much. Fantastic. All right, let's do it. So Delfi Diagnostics is one of the leading liquid biopsy companies working in early detection of cancer and therapeutic monitoring, and using a novel approach for cancer detection. So in the next slides, I really want to discuss cell-free DNA fragmentation technology. Is it the founder of the technology founding on the Delphi approach, and I want to talk about how it can be used to actually quantify tumor burden in cancer patients that are going through systemic therapy. I will go through the development, training, and validation of the Delphi Tumor Fraction model, that is an assay based on cell-free DNA fragmentation to monitor treatment response and detect molecular response. So, and I don't need to go into details about liquid biopsy, the potential, and because of the great presentation from Kaitlin and Amanda today. So thanks so much for the intro, but we can dive a little bit deeper into cell-free DNA fragmentation technology. So the fragmentation approach is a next generation liquid biopsy method, and it's based on cell-free DNA sequencing. It offers a higher signal to noise ratio compared to first generation methods, and tending to be lower in cost and more widely applicable for cancer screening and monitoring. As the main goal for Delfi Diagnostics is as actually early cancer detection and into lung cancer screening right now. So the first generation liquid biopsy approaches, they're often tissue informed and they require very high sequencing coverage and pretty expensive. So the second generation liquid biopsy, they're like related to DNA methylation or other kind of form of epigenetics signature, and they show improvements in terms of sensitivity, but the costs are still pretty high because of the assay, because of the sequencing coverage. So that brings few problems also there. In contrast, the cell-free DNA fragmentation approach really is able to identify millions of potential differences with much lower sequencing requirements. So the assay that we have is actually a 4x sequencing. And it needs right now only 4x sequencing. It needs less than 1 milliliter of plasma, 1 nanogram of cell-free DNA. So it's a low input, low cost approach that is highly effective for both early detection of cancer and identifying treatment response. So the Delfi Diagnostics, the laboratory really employs a straightforward assay. We extract cell-free DNA from less than 1 milliliter of plasma. There is no need of buffy coat or tissue material, and we use cell-free DNA to generate sequencing libraries and perform whole genome sequencing at 4x again. And the efficacy of whole genome sequencing really lies on the capacity to identify different features within the data, including epigenetics data such as transcription factor binding sites, mutation signature, translocation, deletion, chromosome, copy number changes, so loss and gain, and diverse type of other genomic features. So among these features is a clear, very important one. It's the short to long ratio, which quantifies the length of DNA fragments and compares them to non-cancer reference samples that we generated over the years. So that is defined the fragmentation pattern across the whole genome. So the fragmentation is a fundamental part of the data, but that is actually one of the features that we have in the whole genome sequencing data. So, and given the high sensitivity, low volume opportunity that Delphi using the Delphi approach, there is a disruptive opportunity not just for cancer screening, but also for monitoring treatment response and detecting residual disease. So in this presentation, I will discuss heavily about how the genome-wide fragmentation approach can detect treatment response and overall survival. So this specific assay is actually called DELFI Tumor Fraction, and is able to quantify circulating tumor burden, circulating tumor fraction using cell-free DNA. And the DELFI Tumor Fraction, or DELFI TF, is a tumor and mutation independent approach for cancer detection and monitoring. It is actually a pan-cancer assay that it works in different cancer types and requires, again, less than 1 milliliter. So I'll show you data that the Delphi TF data, Delphi tumor fraction, has very high correlation with mutation data, with over 96% of correlation. We have data that anticipates imaging, anticipates races in terms of treatment response. It costs way less, and it's tumor and therapy agnostic. So it works on colorectal, lung, liver, pancreas, breast, melanoma. So everything that we work with, it's really able to capture. And it is a valuable pharmacodynamic monitoring tool, and it's really used in phase 1-2 trials, informing for drug activity. So we have ongoing collaboration with several innovative biotechs and big pharma to assess the treatment monitoring using Delphi tumor fraction. So, and here let me go a little bit into the also drug development challenge that there is. It's well known that in the drug development path, moving to next clinical development stage, there is a standard success measurement. And the problem that there is always a lack of efficacy between phase 2 to phase 3. So around 70% of phase 2 trials do not advance. And that's because what happens before in phase 1 didn't have any kind of efficacy there. So the approach that DelFi-TF has is able to give molecular response data into phase 1 trials and able to improve the phase 2 success rates. Excuse me. So here let's dive a little bit more into the approach of Delfi Tumor Fraction and how we really can able to quantify molecular response. So in the monitoring program at Delphi, we have presented several data and cohorts of different cancer types treated with systemic therapies. For today's presentation, I'm going to describe the cohorts and data that have been published. And the first cohort that was processed is a metastatic colorectal cancer patient cohort treated with chemotherapy, which internally we call Cairo-5 because of the clinical trial. So that was the original cohort that we used to develop and train the Delphi tumor fraction algorithm. And this cohort was actually published, the data were published in Nature Communication in 2024. So all the next cohort, they were actually used to validate the model. So we developed in the metastatic colorectal cancer Cairo-5, and we can validate it into every cohort that is coming through. So the DelFi-TF model is a locked and validated assay that can be applied to all new cohorts arriving. So the next cohort, the next large cohort that we have is a lung cancer cohort treated with immunotherapy. In this cohort, we validated the DelFi-TF against a deep target sequencing 500 gene panel for point mutations. These were very important to validate the sensitivity against a highly sophisticated deep target sequencing approach. So the next two cohorts are also on lung and multi-cancer that were more to validate the DelFi-TF against imaging. So to identify how early the DelFi-TF can peak like molecular response or progressive disease earlier than imaging. And we know that liquid biopsy has an advantage over imaging. So and now let's focus on the first cohort, that is, Cairo-5 cohort published in Nature Communication. So this cohort is around 150 patients diagnosed with colorectal cancer with metastasis in the liver. We obtain the plasma and we process the Delphi assay. So upon immediate observation of the fragmentation data, we notice a very high aberrant profiles in the baseline sample. So this top data that you see, this noisy data, is really coming from baseline pre-treatment samples. And interesting, and let me step back. So this plot is actually how we plot genome-wide fragmentation data, and the x-axis is the chromosomes. From chromosome 1 to 22, there are different arms, and all this every line that you see in every facet in the plot is actually one sample. So normally, when a non-cancer sample would be like very flat across the genome, because it would be diploid, there wouldn't be any aberration, and it wouldn't have any fragmentation aberrancy. So here, this noisiness in the data really show like cancer presence. And this method has been incredibly validated against clonal hematopoiesis, against germline. So any issue that coming from white blood cells or other non-tumor related phenotypes, they are not identified by fragmentation. So it means that we do not need any buffy coat or any tissue to identify cancer presence. That's simple. So in the noise that we see in the data, the aberrance profiles that we see in the baseline pre-treatment samples, these are gone away when there is treatment. And you see that the profiles in the responders when there is partial response, there is the samples look very clean. And it's interestingly, when the noise of the data, the aberrant profiles, is actually come back in stable disease and in progressive disease. You can see very aberrant profiles here. So, and after seeing these results, we realized that we could build a model able to track a response to treatment using only fragmentation data. And this cohort was perfect for this because every patient had the tumor genotype for specific mutations. We had KRAS, NRAS, and BRAF. So we knew exactly which patients had tumors with these specific mutations. So we isolated the cell-free DNA from plasma. We performed the DELFI assay. We performed digital PCR on KRAS, NRAS, on BRAF. So we ended up with a large data set where we had fragmentation data from all samples, but also we had the exact mutant allele frequency, so the tumor burden, the tumor fraction for every sample. So we basically built a random forest machine learning model that we have obtained and we call DELFI Tumor Fraction, and right now is able to quantify tumor fraction as good as digital PCR does. So, but only requires 4x whole genome sequencing data. There is no need of tissue, there is no need of buffet. And here on the right, you can see the correlation plot between DELFI TF on the y-axis and MAF on the x-axis. And they have a very good correlation, about 0.92, so it's basically a perfect correlation. And there are some cases, very interesting cases, where samples are actually MAF is 0. So on the left of the plot, there are red dots. You can see they're 0% MAF. But if you look at the DELFI TF values, there are like samples with above 1%, with 5%, 10, 15% of ctDNA. So this is very high tumor fraction, and it cannot be, it is no noise. We know it's tumor presence. And the key thing that mutation sometimes, digital PCR, is not able to capture those samples because different reasons. Digital PCR may be failing as an assay, or there is a clone switching in the cancer. So there are multiple explanations for this. But if you have a whole genome sequencing assay, as also Kaitlin was showing us today, you really have an ability to identify different genomic regions with potential differences, potential cancer presence. So the whole genome sequencing has real ability to identify tumor presence even in those cases where there is dynamic happening and you don't see any more specific mutation. So we got the DELFI TF model and we applied to the whole Chi-5 cohort. So we could quantify exactly what we were observing by eye in the fragmentation profile that I was showing you before. So you can see this non-cancer reference, they're part of the study, they're basically close to 0. You can see the samples collected at pretreatment, so at baseline, they have a very high tumor fraction, extremely high. So these colorectal cancer patients that have high shedded tumor, very high tumor fraction. So samples that have actually collected during treatment, they have a very low ctDNA. So there's a big reduction of tumor burden, and you can identify the reduction in DELFI tumor fraction. But when there is progressive disease identified by imaging also, DELFI TF detect is increased. You have a way increase about that. And what is important to point out is that DELFI TF allows the ctDNA quantification in both the mutant cases, that the red box, against the wild type cases, that is the pink box. So you can see that basically the values between patients, they're pretty similar. And that is important for us because it really confirms that we have a tumor and mutation independent approach that quantify tumor fraction. So we can apply that to different cohorts, to different, regardless of the mutation, we can identify tumor fraction. So we applied the DELFI TF to the clinical data in the whole cohort that we observed. The DELFI TF is a good prognostic factor to determine which patients they have a better treatment response. So low DELFI TF are going to have a better survival compared to the high DELFI TF. And that is true also for MAF. So liquid biopsy is really able to identify those patients well for ahead of treatment response. So here I want to give you a quick example of a patient. These are imaging data, again, published. So this is a patient that had high liver metastasis at baseline, and these metastases go away during treatment and they come back later on after several weeks of treatment. So, and you can see that the lines, between Delfi TF, it's really rising up earlier than imaging, showing that there is, identification of treatment response and progressive disease earlier than imaging. And this is one case. We have way more than that, and I'm going to show it to you also in a second. So and now that we have a model that quantifies tumor fraction, we want to measure the performance metrics and to test how it works also in other cohorts. So and again, these are locked model and apply to new cohorts. So the largest and most important cohorts that we have is lung cancer cohort treated with immunotherapy. And this we validated the Delfi TF against a deep target sequencing panel. And in the other cohorts of lung cancer, we correlated the Delfi TF against imaging. So given the alignment between imaging in this cohort. So this is also was presented at AACR 2025. So I'm happy to share the slides of the presentation by our JHU collaborator. So in this cohort of stage 4 lung cancer, these were treated with immunotherapy, and we collected plasma and buffy coat longitudinally, and we processed in parallel the Delphi assay and a deep target 500 gene panel, so as a deep sequencing panel. And then in terms of logistics and requirement, they are completely on a different scale. So there were around 700 samples, and I think for the Delphi assay, we performed like in around 3 weeks. For the MAF assay, I think it took us like 6 months to get the data back for everything. 4 milliliters requirements against like less than 1. So the logistics are so different. And we process the buffy coat to filter out germline and chip mutants for the MAF. So we make sure to have a maximum MAF. So we code both Delfi TF and MAF for a correlation analysis. And here, what it looks like a correlation analysis, we expected a good overall correlation with MAF time points across the whole cohort. These are both baseline, the first plot on the left, so collected pre-treatment, but also at the landmark time point. So the landmark time point is a clinically meaningful time point collected between 3 and 9 weeks. And this is really important time point because it's really important to identify treatment response. So we actually look at the time point, looking at Delphi tumor fraction. We're looking at the patient that have they're positive for Delphi tumor fraction at the landmark time point compared to the one that are not positive. And you can see that and we call them the positive, we collect the molecular progressors. The one that they're not positive, so the negative, they are molecular responders. So you can see by the KM plot how the patients that identify as molecular responders by Delfi TF have a way longer progression-free survival compared to patients with a molecular progression determined by Delfi TF. And this is a very interesting analysis that I really talk a lot to collaborators because it's a one-time point analysis. It's not dynamic. It's a specific between 3 and 9 weeks, depends on the therapy, but it's really powerful because you don't look at dynamics, you look at one time point. So I think it's really meaningful clinically and logistically kind of in the real world setting. So at this point, we want to evaluate the detection percentage for both Delphi tumor fraction assay and the mutation approach. So we specifically focus on a series of patients that had also tissue exome sequencing. So we could be sure to track the tumor-specific variants with a mutation approach. So we had plasma data, we had buffy coat, we have tissue exome sequencing. So, and here I'm showing you the detection rate in the for the MAF and the Delfi TF assay. So you can see when there is a detection, there is a very high detection rate for the MAF when you use only plasma sample, when you only use the plasma data. But if you use plasma and you filter out buffy coat and you look only the variants that are in the tissue, the detection rate goes below 60%. So what it tells us that is a lot of these variants are actually not coming from from the tumor itself. They are something else. They are not tumor derived. So, and this is, pretty incredible, but it's, this is, we know that this, clonal hematopoiesis, germline mutation, they make a huge impact. So you need to have, tissue, as, also Kaitlin was showing to us. But the genome-wide fragmentation approach, since it doesn't need buffer, since it doesn't, it's not confounded by clonal hematopoiesis and germline, you can see that the detection rate is above 65%, is above actually tissue-specific mutation detection rate. So that suggests that the sensitivity of the genome-wide assay is better compared to, single variants or mutant allele frequency approaches. So, and this is on analytical side, the detection, but worried about the clinical sensitivity. So we look at the clinical sensitivity, we notice a discrepancy between Delfi TF and MAF, and that's what I'm showing you here. So we investigated specific samples and patients that were discordant between Delfi TF and MAF. So these are a total of 48 patients that were discordant between whole genome sequencing and MAF. And on the top, these red lines, these are samples detected as molecular progressors by both Delfi TF and MAF. So both the approaches, they are concordant in these patients. But the bottom patients, they are detected as molecular progressors by MAF, but they are called response. They're called as molecular response by TF, by Delfi TF. Okay, so these 20 patients that only Delfi TF is telling there is a good response to treatment. Okay, so to understand, which method is actually it's more related to the actual clinical data, to actually tumor burden, we look at the clinical data. So, and here we show you the KM plot. We look at the clinical data, observe that patients detected as molecular response only by Delfi TF have a way longer overall survival compared to patients detected as progressors. And these differences was found not only by Delfi TF and the whole genome, being a whole genome sequencing approach. And all the patients, other patients that detected as progressors by MAF, and MAF called them as, the same, the one on the red, the one on the blue, top and bottom, they are the same for the MAF approach. But if you look at the Delfi TF, it's going to tell you which patients are actually responding. So it's really powerful. So here I want to give you for like last 30 seconds overview of a study that we've done on a multi-cancer cohort treated with immunotherapy. This is a study under review in Science Translational Medicine. So in this cohort, we apply the Delfi Tumor Fraction against imaging, and we look at week 10 of immunotherapy, how Delfi TF and imaging they were correlating. So here the KM plots on the left, this is imaging, so dividing the patients by imaging, so partial response versus progressive disease. There is no separation because at week 10 imaging is too early to identify. But if you look at the middle KM plot, you can see a huge separation between the molecular responders identified by Delfi TF versus molecular progressors, again, identified by Delfi TF. So Delfi tumor fraction at week 10 is really able to identify the patient that are responding to treatment. And while imaging is not even close to do that, it's too early. And if you look at the KM plot on the right side, these are patients called as stable disease at week 10. So you don't really know, anything about stable disease in imaging. So what about, liquid biopsy and Delfi? So there is clearly a separation between the ones that are called as responders by the fragmentation approach versus the one that are not responders. So again, liquid biopsy is a huge opportunity for monitoring treatment response and quantifying molecular response and in cancer patients. So here I get to the conclusion. So Delfi Tumor Fraction is a monitoring tool for portfolio management in stage 4 metastatic cases. Is a cost-effective alternative to mutant allele fraction and with low requirements and tumor and treatment independent, and with strong data that correlates with MAF and strong data that anticipates imaging. Thank you so much, and happy to take any questions.
Maybe one question. Can you hear us?
Yes.
Okay, so will he hear us? Okay.
Yes. Yes.
Okay, Lorenzo, can you hear me? This is Chris.
Can you hear Chris?
Oh, great. Okay. So great talk. And I guess I had a question. I don't want to get too far in the weeds here, but I think it was the second lung IO study. It was a smaller cohort that was presented at AACR, I think, where you had 48 patients and you were showing that the Delphi response, the Delphi tumor fraction was a better predictor of response than MAF. And I guess I wanted to try to understand, because I think that was at the landmark time point of 3 to 9 weeks. You were looking at both the Delphi fraction and MAF. And what were you actually looking at there? Maybe you can explain that, because were you looking at clearance by, so no ctDNA detected by the Delphi fragmentation approach versus MAF, or what was it that you were looking at, and why do you think that the Delphi approach was a better predictor of response?
Of course. Well, thank you so much. Thank you so much for the great question, Chris. And that's definitely very interesting, and we're fascinated about the results. What I can tell you that first, the landmark analysis, it's only it's one time point, and it's based on detection of ctDNA, yes or no. For the DELFI tumor fraction, we have a threshold. This is our threshold for calling detection, yes or not, using the Delfi TF. And for MAF, it was if it's positive or negative. If it's if it's above the limit of detection identified. So yes, that is basically positive versus negative. We tried different approaches, of course. You look at dynamics, but what we believe the landmark analysis is really meaningful if you have only one time. And if you look at one time point only, because in several cases that we did, collaborators come to us, and even the clinic, there are often cases where there is no pretreatment available, and because it was not collected, because it's not there, so we needed like an assay able to quantify a tumor fraction, using one time point. So that's what we've done for the analysis. It's a landmark analysis. And again, why the fragmentation, the genome-wide approach is more clinical sensitive. I think it's really relies on the fact that it's not confounded by clonal hematopoiesis. It's not confounded by other variants that are not tumor derived. And we see a lot of mutation artifacts in the data when you really sequence deep with mutation data. So the fact we see variants at 0.2%, 0.5, 0.8, it depends, that they cannot be identified in the tissue. So if they're not identifying the tissue, are they tumor? Are they not tumor? So I think it's the fact that it's not confounded by clonal hematopoiesis and other factors. That's really, and especially because of patient undergoing different type of therapies. You have a lot of new mutation, new variants happening during treatment that actually you didn't have even before. Sometimes you don't have even the tissue. You have only those variants when in the plasma, but they're not tumor. They're coming there, like the white blood cells get mutated also as the patient is undergoing like systemic therapy. So there is some of that is really confounding like heavily the mutation there.
Question, but that's okay. I think we're already way over time, so I'll follow up with you later, Lorenzo. That was very very nice talk. Thanks.
I appreciate it. Thank you, Chris.
Folks, I don't know if it's Lorenzo, can you hear us?
Yes.
Yes. Okay, great. Well, thank you so much. Apologies for all these technical difficulties, but thank you for staying with us and for coming. And the folks online really appreciate that. And I would like to thank our amazing speakers today. Again, thank you. Thank you so much. And then I also would love to acknowledge our co-organizers and the sponsors for the breakfast. It's the Frederick County Chamber of Commerce and Maddie and Abby. So thank you so much for being here and for providing us this yummy breakfast and for supporting the event. And then, so our next Biotech Connector is going to be in November 20th, and it's going to be in focused on the cell and gene therapy. So please, attend, kind of virtual or come over here. Thank you so much. Apologies again. Thank you. Thank you.